Hey guys, good morning. This is morning 4 a.m. and Indian Standard Time. I'm in India. So why I am recording the video because after having like conversation with you over the Zoom meeting and request from several of my students, I just am releasing this video to know how to make the notes, right, or the charts for the pathways. So I'm going to do it for all the pathways one by one slowly. First, we will start with the glycolysis. I don't know how much this is going to take time, so you can just skip forward the video, or if you really want to learn even the glycolysis, you can continue watching as per your pace. But the main goal of this is how to have one pathway on a piece of a paper with most information, more relevant information on the paper, and that paper we are going to stick on the wall of our room so that when we are coming out and in and out in the room, right, at least once a day we can look at the pathway until unless we memorize everything related to the pathway, right? Because then we will have this pictorial memory of that paper in our brain that will be helping us in the exam. Second thing is we may leave some information right now today in this video, but as soon as like you come across a relevant information of the pathway, related pathway, you are going to write it down on that chart so that in the end you have a complete big chart in which you have all the information. Number two, on the screen, I'm going to write the bigger picture, bigger font size. I'm going to use it the zoomed version, but you are going to write in a smaller font size, okay? Because remember, you have to squeeze out everything on a one single piece of a paper. So first you can have, like for the first, this is the first video, you may have a hard time to adjust the arrangement, but soon you will be getting a hang, like used to it, and you will be getting a fairly good amount of an idea, right? Or in the end of the video, you can just skip the video to the end and see how the entire page is divided into various parts. And then you can really skip back to the beginning and start making it. Because in the end, on a piece of a paper, you will having an idea how I drew it. So this is the first video, so I'm really not sure how it's gonna go. So I hope you will bear with me with that. Okay, so I'm gonna share the screen with you, right, of the page which we are going to use. And this is a page we are going to use to draw which cycle, the very first cycle, like a baby cycle, the glycolysis. So this, see, I have zoomed in, right? So for you, this is a piece of a paper. So hopefully you are going to divide it into two halves. One half we are going to use to write the cycle. The other half we are going to use to write the relevant information. If you run out of the page, use the back side of a page or use another page. So you can stick two pages side by side. That will depend upon the information about the cycle, right? So let's begin about the glycolysis cycle. Use a smaller font size. I'm using the bigger one. First of all, what should we be writing about the cycle? Remember, I told you first you will be writing where the location of the cycle is. Glycolysis is a cytoplasmic or cytosolic cycle. It takes place in the cytoplasm. Whether this is a catabolic or anabolic cycle, this is purely a catabolic cycle. Catabolic cycle. What is the starting substrate? Substrate is glucose. It starts with the glucose, and the end product you get is, you can see you are getting a pyruvate, or sometimes you may say in anaerobic conditions you get a lactate to it, right? And this is what this is, catabolic cycle. Catabolic cycles all are energy producing cycle, right? All catabolic cycles are energy producing cycle, whereas anabolic cycle that we will see in gluconeogenesis is energy releasing, energy consuming cycles, I'm sorry, right? So this is basically, we should be writing it out, and then in the end we will be writing how much ATP you see, right? There are 9 steps of the cycle, but you will see the 10 substrates that are involved in the cycle. And first starting point is, what did I say? It is glucose. Glucose is the first starting point, and we convert glucose into glucose 6-phosphate. We are adding a phosphate, right? First, I'm going to write the skeleton, right? Glucose 6-phosphate to fructose 6-phosphate. Glucose is an aldehyde, fructose is an keto. So they are brother sister to each other. So if we have to convert them, like interconvert them, that is a reversible reaction. Isomerase can help us because these are just the aldose and the ketose version, right? So fructose 6-phosphate, that is just having what? How many phosphate? Just one phosphate. So we add on one or more phosphate to it. That gives us fructose 1,6-BP. That is B for bis, P for phosphate, right? And then we move on further. Now we have what? A molecule, 6-carbon molecule, right? This is what? This is for your understanding. Don't write it down, okay? You have 6-carbon molecule with 2 phosphates to it, right? Now we can chop it into 3 and 3, and every one component will be having one phosphate, another component will be having another phosphate, right? So what I do now is I'm gonna break it apart, rip it apart. And who is gonna rip it apart? An enzyme named aldolase, right? So we get dhap. This is like we are chopping it off with the sound dhap. Okay, so that is one substrate, dhap, chop it off, right? And another is glyceraldehyde 3-phosphate I get, right? Or difference between these two are, they again, DHAP is an keto version, G3P is an aldehyde version. So they are also brothers and sister, and isomerase enzyme can help them to get interconverted, right? DHAP we cannot use as such. So we will continue the cycle with G3P, glyceraldehyde 3-phosphate. Again, what we are going to do, we are going to add 2 phosphates. We need 2 phosphates on it. So we will be adding 1 phosphate onto it, right? And then what we will get? A molecule G3P, right? And that will be having 2 phosphate, where at first position and the third position. When it is 2, then we call it bis, right? Bis phosphoglycerate, BPG, right? Up to this point, what we are doing, we are adding the phosphates to it, right? And usually phosphates come from ATP, else is inorganic phosphate. Now what we have to do is, now we have done lots of investment. What we need is, we need to get the energy out, because the glycolysis is a catabolic cycle, right? So after this half, what we will be getting? We will be getting energy. So we will be chopping this molecule kacha off, right? And with every chop off, lots of energy is being released in the form of ATP, right? So 1,3-BPG will release the energy, and what we will get? 3-PG, right? 3-PG we get, and there will be a release of energy in some form, right? 3-PG, then 2-PG. We are remembering the first step, right? You can correlate 1,3-BPG, 1 phosphate is gone, 3-PG, then 2-PG. 2-PG is just moving moving the same phosphate group, right? The phosphate group is the same. Earlier it was at 3 position, now it will be at 2 position. That is the only difference, right? And that is kind of a, you can say, just shifting the phosphate group. We are not interconverting isomers, nahi bana rahe. We are not making the isomers. We are shifting the phosphate group, and that is done by enzyme mutase. Okay, so you have now 3-PG. 3-PG will get converted into PEP, phosphoenol pyruvate, very important intermediate, and finally we get pyruvate, right? And pyruvate has 3 fates, and we will see all the 3 fates. One is lactate. We need to know the details also. Another is acetyl coenzyme A, and another majorly we called as alanine, right? 3 fates, and those 3 fates of pyruvate depends upon whether this pyruvate is staying in the cytosol, or it is moving to the mitochondria. That we will take into later half of this video. Okay, so now what we have this entire cycle of the glycolysis on a one piece of a paper. Maybe you are left with a space on the bottom. Just don't worry, we have lots of information to write it, but this you should be able to squeeze it on the single piece of a paper, on the half part of a paper. Right now we are going to fill what the enzyme slowly, one by one, with the understanding about the enzyme. Why I'm doing that? So that you even memorize it for the examination point of view. Glue to glue 6-phosphate. What we see here, what do you see here? You see 1 phosphate group is being added at the 6th position. From where this phosphate is coming from? This phosphate is coming from ATP. Right? Initially, what we do is molecule is stable, it's dead, it doesn't want to interact with something. So you have to charge up the molecule. That is by adding using an energy, right? So ATP is being used, and this ATP is being used. I rarely write what comes out unless it is a special step. ATP is consumed, one phosphate is used. So it means we are left with ADP. So I'm not gonna fill out my pages, because more we write, more we will see that it is congested. So we are going to write the least and the most in the sense for the information, least, rest is understood. Okay, so ATP is being used, you get glucose 6-phosphate. Why we do this step? Now glucose is like, we have the blood transport, it can go in and out very easily across the cell membrane. By having glucose 6-phosphate, it stays inside the cell. Same thing is if we convert fructose into fructose 6-phosphate, it stays inside the cell. Phosphate does not allow it to go outside, because phosphate is adding a negative charge to it, right? So cell membrane is going to push it further. You cannot go out, you cannot go out. Same is the galactose. Anything you make a glucose, like a simple molecule to its phosphate version, it is locked inside the cell. So this is how we do, why the cell do it. First of all, student comes inside the room, right? And then you lock the room so that he cannot run outside. He has to learn the entire chapter. Okay, so that is why this step is taking place. And when we are adding a phosphate, as I always say, what we use the enzyme is to add the phosphate is the kinase. To remove the phosphate is the phosphatase. To add the phosphate, it is a kinase enzyme. You will always see the enzyme kinase is adding a phosphate, except 1-2 places. One of the exception, we will see it now. I don't think that in your course you have any other exceptions. Kinases add phosphate. Remember that. Phosphatases remove the phosphate. So there is some kind of a kinase. What kind of kinase is that? There are 2 kinases, glucokinase, hexokinase. Right? Glucokinase, hexokinase. Why the name gluco? Name is gluco, right? So it has a love or affection for the glucose. Right? It means glycolysis is like most of the hexose sugar, whatever you have in the diet, you can convert them via this pathway. Yes, those all are convertible. We will see eventually with the time. But glucokinase is specifically for the glucose. And when it has what? It has very, very high Km. High Km means Km, what I say, is an affection, love for the enzyme. Right? So if the Km is high, then there is low affinity. This is inversely proportional. Affinity is low. Remember that they both are opposite. They both are opposite. Km is a substrate concentration, right? So if the substrate Km is very, very high, affinity, affection for that molecule is low, right? So for glucokinase to convert glucose into glucose 6-phosphate or to work, we need lots of glucose in the blood. We need lots of glucose in the blood. We need lots of substrate concentration, higher Km, right? So glucokinase only works when you have just eaten, because when you have just eaten, what you have higher glucose in the blood. And that point in time, glucokinase will start converting glucose into glucose 6-phosphate. Hexokinase, the other enzyme, is rather a simpler enzyme, simpler version, very like humble enzyme. Any hexose, it will work. If you find the glucose or the fructose, right, or galactose, it will work. It doesn't mind working with any kind of the hexose. So that hexokinase has what? It has lower Km. We will see how hexokinase and glucokinase are different, right? So I forgot, don't add this information right here, because up here on the second half, we will make a chart. And in that chart, we will learn the difference between glucokinase and the hexokinase. Very important question for the examination point of view. They ask you question in one or another way. Well, coming back to our this part, right? Glucose to glucose 6-phosphate can be done by the glucokinase enzyme or the hexokinase enzyme, right? And this step requires magnesium ion. This step requires magnesium ion. Then glucose 6-phosphate is converted into fructose 6-phosphate, and the enzyme is, these are isomers of each other. So enzyme is isomerase, right? And what that is important, you should be knowing the isomerase, but what kind of phosphohexose isomerase, right? Otherwise, it's an isomerase enzyme, right? Nothing doing in this. And fructose 6-phosphate to fructose 1,6-bisphosphate. What we did, again, we have added one more phosphate to it. So one more ATP is being consumed. So ATP is being consumed. ATP is being consumed, and what you are getting ADP, right? What you are getting ADP, and the enzyme is again what kind of an enzyme? That is an kinase enzyme, because we are adding phosphate enzyme, and a very special enzyme, phosphofructokinase, right? Phosphofructokinase, and that is one. Phosphofructokinase, another part we will see on this half. Right half mein hum likhenge, we are going to write it down how it is different. Fructose 1-phosphate, PFK1, and there is PFK2 also, but there is this what kinase PFK1 that is heading the 1-phosphate to in this. Now fructose 1,6-bisphosphate is gonna getting cleaved into, we are chopping it off, and that chopping is done by whom? That chopping is done by, hold, screen got some issues. Okay, now, right. So where this we were here. So enzyme here is aldolase. Again, there is aldolase A and there is aldolase B. Right? There are two versions of the aldolase A, aldolase B. With the glycolysis, we will see there is always aldolase A. Remember, A comes first, right? And very first cycle we learn is the glycolysis, right? Another B version, aldolase B version is with the fructose. We're going to write down on the right half later. But aldolase A is being converted, is chopping it off. We have glyceraldehyde 3-phosphate. What else you see? But now here what we see is there is one more phosphate that is being added. So are we going to use the 1 ATP? No. At this point, the contribution is done by PI, that is inorganic phosphate, right? Inorganic phosphate. And whenever you have the inorganic phosphate added to it, right, then that is sort of an substrate level phosphorylation. This step is known as substrate level phosphorylation. And the beauty of this is there is a dehydrogenase enzyme that is being involved here. And that too is substrate kya hai aapka? Substrate is glyceraldehyde 3-phosphate. So enzyme name is glyceraldehyde 3-phosphate dehydrogenase. Very important examination point of view kind of an exam. Okay. Whenever we have dehydrogenases, right, those who have already went through the enzymes chapter, they know about the enzymes. There is an oxidation reduction that takes place, right? There is an oxidation reduction that takes place. What this cycle is about? This cycle is about, this is catabolic cycle. So energy should be released, right? Energy is in the form of ATP, GTP, right? And another form of energy in the reducing power form of energy is NADH. So you are gonna release one NADH. If let's say you don't know NAD positive is gonna release or NADH is gonna release, remember this is a trick. This is a catabolic cycle. So NADH you are gonna release, because NADH, when it will go to the further cycle in the electron transport chain, it will be getting converted into energy, ATP, right? And NADH is equals to 2.5 ATP. NADH is equals to 2.5 ATP, right? And another form of reducing form is FADH2 is 1.5 AT. Since this is the first cycle, we are gonna take longer so that you know how to write it, and then further cycles I'm gonna speed myself up. Okay, so you have 1,3-BPG, right? And you are releasing what? NADH. That is another first energy thing that you see. Now 1,3-BPG gets converted into 3-PG. So what is being ATP, one phosphate is being removed from here. And when you remove one phosphate from here, right, what you release, now this is the first ATP you see here. This is the first ATP you see here. And since we are removing phosphate, so what we will expect? We will expect a phosphatase enzyme. But this is an exception. Remember, I told you one exception you will see in which the phosphate is being removed, right? But the enzyme is not a phosphatase enzyme, but rather it is, what is the name of the product? 3-PG is the name of a product. Name of the enzyme is 3-PG kinase enzyme. So this is the exception which I was talking about. I don't think in your course you are going to see any other kinase doing removal in which it is seeing that the phosphate is being removed. So remember this enzyme name, right? So 3-PG is converted into 2-PG, right? The phosphate is being moved from third position to the second position, and the enzyme is a kind of a mutase enzyme. What kind of an enzyme is a mutase enzyme? Then 2-PG is converted into PEP. Again, one phosphate is being removed here. So what we are releasing? We are releasing one ATP molecule here. And the enzyme here is enolase enzyme, right? Right? Eno, remember with this eno, right? Eno is related to peptic acid, right? Just remember that that is kind of a gastric acid, right? Peptic acid is related to the gastric acid, right? And then we use what we are eating. We are eating eno. So enolase enzyme is giving the PEP, right? Phosphoenol pyruvate, and one ATP is being released, and this PEP is converted into finally into pyruvate. Your very key important enzyme, right? And the enzyme here involved is another important enzyme. What is the name of the product? Pyruvate, right? So enzyme name is pyruvate. Okay, PK, right? Movie, remember the one name, movie name PK, right? So this is the PK enzyme, and that is very important, and that is going to help us make pyruvate. So these are all the phases. So I'm gonna zoom out a little bit so that I can add the notes. What kind of a notes? See the first half of the cycle. So this half of the cycle, we are utilizing the ATP, right? So this is what we call it as investment phase. You can write it down. This is an investment phase. Okay? And initially you do the investment, and finally what we are getting after this, we are getting NADH. Is that true? Right? So this is a payoff phase, right? Or you can say what it is called as, right? This is energy producing phase. And what is the beauty of this phase? You get one ATP, two ATP actually at two places, and you get one NADH. Write it down, write it down. And that is times 2. Why that is times 2? Because the DHAP, this DHAP will also get converted into G3P, and then again you will make pyruvate. So this half of the cycle is times 2. So you will be getting 4 ATP and 2 NADH, right? So since we are doing the energetics, we just remember freshly the energetics, just write it down. Okay, so what we are doing is we are releasing 4 ATP and 2 NADH, right? This is our add. 2 NADH is 2.5 ATP. So I can say 5 ATP. Can I say 5 ATP? And what we have consumed in the upper half, we have consumed 2 ATPs, right? So if you do the maths, 4 plus 5 is 9. 9 minus 2 is what we are left with. We are left with 7 ATP. So one glucose molecule gives you 7 ATP. That is a general thing. You have written it down here on the right hand side. ठीक है? अभी ये हो गया है. This is about the energetics in general. it is going to change about the anaerobic glycolysis also, right? It changes. So this is the one thing. And now what we need to know is glucokinase and hexokinase difference, right? What is the difference between glucokinase and hexokinase? Before I forget, I want you to know that only there are 3 steps which are irreversible, which you cannot revert, because there is gluconeogenesis in which we synthesize the glucose. So many a times, this is another important question. Is gluconeogenesis a reversal of glycolysis? No, it is not, because you can use a highlighter. You can use a highlighter, or you can, if you don't have a highlighter, right, you don't have to rush and go buy stationaries. You can just write 1 here, 2 here, or those who are using the highlighter, they can highlight it, or just write 1, 2, and the very last step here, very last step. Okay, what are these highlighters? 1, 2, and 3, right? In the bottom, write it down here, *1, *2, *3, right? Or those who are using the highlighter, so just make a highlighter kind of a thing. What do we mean by that? They are what? They are irreversible steps. They are what? They are irreversible step. So now this part is done. Now coming back to very important question, hexokinase, glucokinase. Now you know the pathway, where it is occurring, catabolic pathway. Now you have investment phase, payoff phase, 7 ATPs are being produced, NADH, which step is taking place. You know basically everything. The very first important question, another, is what is the difference between hexokinase and the glucokinase, right? So glucokinase, as the name is suggesting, what we are using, it works with the glucose only. It do not work with, it has nothing to do, no interest at all with the fructose or any other hexose. Glucose is my first love, and the like just ना 1 lady person, ग्लूकोज को सिर्फ एक ही लेडी से प्यार है, वो ग्लूकोकाइनेज को सिर्फ ग्लूकोज से प्यार है, इसके अलावा वो किसी को एंटरटेन नहीं करता. It has what? High Km, high attitude है ना, सिर्फ just glucose only. I don't have any other choice, right? I just want sort of high attitude. Attitude is the Km, remember that, right? That makes your life easier. High Km. High Km means there is low affinity, low affection. Attitude है, affection कहाँ से होगा, right? So low affinity, right? So if Km का concentration ज़्यादा होगा, only then it is gonna work, right? And then this is an inducible enzyme. Inducible means somebody can induce it. Who does it? Insulin does it, right? Because when we have lots of glucose, insulin is going to release from the pancreatic cells, right? And then it is going to induce the glucokinase to start working. Otherwise, वो सोता रहेगा. वो उसको बताएगा knock glucokinase start working. Okay, and approximately more than 100 milligram per deciliter of glucose, you can write it down, must be available to, right? And that is available in the well-fed state. And where it is found? It is found in liver and the pancreas. Liver is the major site, no? Liver is the major site. So these are the 1 point, 2 point, 3 point, 4 point, and the 5 points for the glucokinase enzyme. So what is the opposite for the hexokinase? Hexokinase acts on all the hexoses. It acts on all the hexoses. I'm going to zoom in, but you are going to write it in a smaller font size. Other hexoses other than the glucose are what? कौन-कौन से hexoses हैं? Galactose, right? Galactose है, mannose है. Support डालेगा, सबके साथ काम करेगा, right? Very cooperative. So low attitude. Attitude is Km. Low Km है, तो high affinity. Affinity means affection बहुत ज्यादा है इसका, right? And it means it works at low blood glucose पे भी काम करेगा. Low blood glucose पे भी काम करेगा, right? And it is inhibited by, this is inhibited by glucose 6-phosphate. Means glucose 6-phosphate क्या है? Go back to the cycle, see, देखो साइकिल में क्या है, हम बना क्या रहे हैं, ग्लूकोज इज गेटिंग कन्वर्टेड इनटू ग्लूकोज 6 फॉस्फेट, ठीक है ना, दैट इज अ प्रोडक्ट. सो प्रोडक्ट इनहिबिट्स द enzyme itself. It inhibits the enzyme. That is a feedback, negative feedback regulation for the enzyme, right? So low blood glucose level पे काम करेगा, और where it is found, it is active in all tissues. It is active in all kind of tissues, not just liver or the pancreas, सब जगह काम करेगा, right? So this is about the enzymes. What is the difference between the enzymes, right? Now, at two places we added here is the substrate, or not two places, but this is your substrate level phosphorylation, right? Is there something else? Let me, right? We need to know about aldolase, right? We need to know about aldolase now. First, now we will be writing about the aldolase. Aldolase A is there, aldolase B is there, right? Aldolase A is almost found everywhere. It is found everywhere. Glycolysis is happening in all kind of the cells. So we are going to stick with the A is sticking with the glycolysis, because that is the primary cycle. We are going to remember it that way. B works with the fructose. Fructose pathway, you will see there is a disease, fructose intolerance. Fructose intolerance is a disease that is related to the fructose metabolism, right? So at that point in time, you can make a connection. I'm going to give you this mnemonic. I'm going to give you this mnemonic, FBI, right? Fructose intolerance is B. That means that point in time, there is a defect in aldolase B enzyme. Very good. Now, another important aspect of this glycolysis is, since this is PFK1, this is your rate limiting step, and this is a rate limiting enzyme of the glycolysis. Very important question for the examination point of view. Rate limiting step and rate limiting enzyme. And I'm telling you, there is two isomers. There are two isomers of the phosphofructokinase. That is for phosphofructokinase 1 and phosphofructokinase 2, right? So there are two isomers of that. And let me see. So what happens here is, we are again going to write it down on the right hand side tab. Okay. You can squeeze in the way you like. Now, fructose 6-phosphate to fructose 1,6-bisphosphate is the case, right? And the enzyme involved here is phosphofructokinase 1. Phosphofructokinase 1. Now, there are conditions in which fructose 6-phosphates get converted into fructose, not 1,6-phosphate, but fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate, it gets converted into, right? And then this fructose 2,6-bisphosphate gets converted into fructose 1,6-phosphate, right? So as you can see, this is 2 and this is 1. Right? It means we are changing the position of the phosphate. In this molecule, the phosphate was at second position. Now, in this molecule, the phosphate, it has 1 position, right? Now, the enzyme here is, involved here is, what kind of an enzyme that is involved here? When we move just the phosphate group, remember here also we moved what? The phosphate group here. That enzyme is a mutase enzyme. So there is this mutase enzyme which is going to shuffle it from 2,6-bisphosphate to 1,6-bisphosphate. But the first step, when we are adding the phosphate group to it, there is another isomer form of PFK, that is PFK2. And how we are going to remember here is, look at the pink highlighter. When you have two isomer, PFK2 isomer, you will be making fructose 2,6-bisphosphate. Whenever you have PFK1, you are making fructose 1,6-bisphosphate, right? This is a normal way of the cycle. This is a normal route of the cycle. This route of cycle is not like a part of a glycolysis, but a side, like other part of a glycolysis. Why that is happening? Whenever you have the very like high elevated concentration or PFK, who is gonna trigger PFK1 to work? This is an inducible enzyme. Who helps it to work? It gets converted into fructose 2,6-bisphosphate, and this substrate activates PFK1 so that it works. So this is kind of a love triangle here, right? And the beauty of PFK1, since PFK1 is our rate limiting enzyme, and that is an inducible enzyme, right? So remember, it is activated. Positive, right? Positive means it is activated. Who is being activated? Fructose 1, PFK1 is being activated by whom? By fructose 6-phosphate, right? If the substrate concentration is high, this is being activated. It is activated by fructose 2,6-bisphosphate ka concentration, क्योंकि बहुत सारा much of the fructose 1-phosphate will start converting to fructose 2,6-bisphosphate also. That is again is triggering the activity of the PFK1 isoform. Okay, so these are the positive like regulators who are going to tell, okay, work fast. And if there is a high ATP concentration, high energy indicator, that inhibits the PFK1, and there is another, if citrate is available, that also inhibits the PFK1, that we will see in the TCA cycle. Citrate we will see in the TCA cycle. So this is about the enzyme PFK1. This is the rate limiting enzyme. This is a rate limiting step. This is irreversible. There is a right hand half to it where you form fructose 2,6-bisphosphate. Isoenzyme involved is fructose PFK2, right? That regulates this step. So this is one regulation point I wanted to give you, right? Now coming back to the cycle again, what is the cycle? Another point that we need to know up here is 1,3-BPG, right? 1,3-BPG is the step what you have here. It is doing here. There is a 3-PG kinase enzyme and you form 3-PG. What it is about, okay? So one more point you can add on the right hand side half. What you will write here is there is 1,3-BPG of the glycolysis cycle that is getting converted into 3-PG, right? Now there is another love triangle here. 2,3-BPG gets 1,3-BPG gets converted into 2,3-BPG or 2,3-BPG, whatever way you want to write it down. Okay, so there is another love triangle that what we study where mostly that we study in the unborn baby, still in the womb, right? For the fetus, because when the lady is pregnant, the fetus wants more of the oxygen, fetus wants more of the glucose from the mom, and that is regulated via this step. Same thing happens when we are at the higher levels. When the oxygen supply is low, then body forms 2,3-BPG. So what it does is it reduces the affinity of the hemoglobin towards the oxygen, and the hemoglobin releases most of the oxygen. That we will take in the study more detail in the oxygen dissociation curve, how it is working. For here, know this, there is another rerouting of this, and the enzyme here involved is mutase, because 1,3-BPG is to 1 se 2 par jana, right? That is a mutase thing. Specifically, you can call this enzyme BPG mutase, right? and then from 2 से 3 pg jana, means one phosphate is being removed. Here what we were seeing, there is a release of ATP, but in this here there is enzyme, that is BPG, who removes the phosphate. Phosphatase removes the phosphate. But important point to notice is there is no ATP. There is no ATP if we take this route. So they can ask you question, person is at higher elevated level, what will be the net energetics of the glycolysis? So 1 ATP you can reduce now, rather than having 7 ATPs, what you are getting? You are getting from the entire one glucose molecule, what you will get? You will get one less ATP. Only 6 ATPs will be produced from one glucose after it is completely oxidized into carbon dioxide and the water. Okay. So there is no ATP being produced, and this pathway is known as what? This is your Rappaport. This is your Rappaport-Lubrin shunt. How to remember this name? I can tell you my stupid way to learn it. Lubrin words come from the labor. It means we are talking about whom? We are talking about a baby here. B is baby, right? Fetus we are talking about, right? So this is 1, 3. Here you can write it down that we can you can correlate with your oxidation ODC curve when we will talk about this, those notes, right? 2,3-BPG. 2,3-BPG helps in unloading of oxygen from hemoglobin to tissues, right? And it contains what? It contains phosphate, and the phosphate is important for that. We have phosphate buffer, so it is important for maintaining buffering capacity, to maintain buffering capacity of RBCs. Very, very important points I'm giving you, highlighted important points I have given you. Okay, so this is about the cycle, right? Let me check if I'm left with something. Now, glucokinase enzyme is done, right? Inhibitors. Okay, inhibitors are left. Who inhibits what? In this cycle, actually, we talk about 2 major inhibitors, right? 2 major inhibitor. One inhibitor is hampering whom? Glyceraldehyde 3-phosphate dehydrogenase. Who is inhibiting it? Iodoacetate. Another one important question from the examination point of view. Iodoacetate inhibits glyceraldehyde 3-phosphate dehydrogenase enzyme, right? And now enolase. Enolase is inhibited by sodium fluoride or fluoroacetate. Sodium fluoride and fluoroacetate halts the glycolysis by inhibiting enolase enzyme. Another very important question from the recall, right? And now there is, if person is exposed to arsenite, then what it happens is, then 1,3-BPG is converted into 3-PG directly. So there is no ATP being produced. It works similar to as like 2,3-BPG wala pathway, but here arsenite is the culprit who does not allow for the synthesis of the ATP, right? So these are the very important inhibitors I want you to know. Now, if I elaborate LDH, lactate dehydrogenase is the enzyme which converts pyruvate into lactate, and this is dehydrogenase, right? So it means there is an oxidation reduction reaction going on, and we release NAD positive. What we are releasing? We are releasing NAD positive. हम release करते हैं. Now you can grab a pencil, right? Like me, I'm grabbing yellow pencil. Now this NAD is positive, this NAD positive is being utilized here. is being utilized here. Glyceraldehyde 3-phosphate dehydrogenase thana, which was releasing 1 NADH. In glycolysis, we get 1 NADH that requires NAD positive, right? So this is the pathway. If you have the lactate, that is being consumed over there also. This is another important question from this pathway. Yes, they have been asked this question in the internal also, and also for the NEET PG. They have been asking these kind of questions. That is why I am making you write. We have done aldolases. We have done aldolases, right? Over here, write it down. Aldolase A deficiency leads to hemolytic anemia. Also, pyruvate kinase deficiency leads to hemolytic anemia. Both of these deficiencies leads to hemolytic anemia. Know this. Hemolytic anemia is by aldolase B, lase A, and PK deficiency. PK deficiency. They both are related to hemolytic anemia. Right. So fate of pyruvate, first thing we have done about the lactate. Now if you are done, your page is full, you can use another page or use the back side of the page. Otherwise, if you have space like in the lower left, if you have space left, you can use that. Mine is left, so I'm going to use it, right? And I'm going to title it Fate of Pyruvate. Right, fate of pyruvate, I can divide into its cytoplasmic fate and its mitochondrial fate. Cytoplasmic fate and its mitochondrial fate. In the cytoplasmic fate, what we are making, we are synthesizing two things. Lactate, which we have just saw, so I'm not discussing it. Another is synthesis of the alanine. Another important question for the examination point of view, right? Alanine can be synthesized directly from the glucose, and this is how we synthesize it. Right, the enzyme involved with this is ALT. ALT is your SGPT. That is your SGPT. We are not discussing that in detail. When time will come, you will see it, right? Or most of you probably have seen it. Now, what is the mitochondrial fate? What is the mitochondrial fate of the? One thing is pyruvate can be converted into acetyl-CoA. Acetyl-CoA is a 2 glucose molecule containing compound. Pyruvate is a 3 glucose molecule containing compound. So this is 1 carbon dioxide is being released. And that enzyme involved here is very, very important enzyme, pyruvate dehydrogenase. There is a short note that can be asked from the pyruvate dehydrogenase. I discuss that also here. I guess I should release a different video with the TCA cycle. This is known as link reaction. Remember this, you mark this. This is a link reaction, right? And this is one fate, and this acetyl-CoA can finally go in the TCA cycle, right? Another mitochondrial fate of this is it can be converted into oxaloacetate by the enzyme PC, pyruvate carboxylase, right? And then it follows gluconeogenesis pathway. Gluconeogenesis is synthesis of glucose. So under starvation condition, pyruvate is routed via this pathway, and this is a mitochondrial pathway, right? So oxaloacetate, acetyl coenzyme A we see in the Krebs cycle, hence it is mitochondrial pathway. PDH and PC is the enzyme that is involved. Lactate is lactate and alanine, that is cytoplasmic synthesis, right? Lactate dehydrogenase or ALT is being involved here. So these are the two fates of the pyruvate. Now that is being done. There is, I told you about what, let's see what else is left. Energetics. So this is very, very important thing. You should be knowing what they are asking. They are asking you about energetics. Whether they are asking you about the energetics of the glycolysis only, or they are about the energetic of the glucose molecule. Keyword matters. Glycolysis ends at the pyruvate. So you have to just count energy up to pyruvate. if you are counting the energy up to pyruvate, do not include NADH, because NADH will be happening after the glycolysis has been done, when NADH molecules are taken down into the in the TCA cycle. So if we are talking about just the glycolysis, we don't talk about NADH, we talk about ATP. So 2 ATPs are being consumed. How many? 2 ATPs are being consumed and 4 ATPs are being released. So net production is just 1 ATP from the glycolysis pathway. The 7 ATP that we have counted, that is when we burn off the glucose molecule to carbon dioxide and the water. So you have to pay attention in the exam, right? And if you have the 2-3 BPG involved, 2-3 BPG involved, then you will do -1 ATP. Then you will do -1 ATP. Am I clear? I guess I'm pretty much clear, and I have given you most of the things, right? And there is very important enzyme, pyruvate kinase enzyme. That is second most common enzyme deficiency. Second most common enzyme deficiency we talk about, right? And more we do it, more things we will come across. If we will come across a relevant information related to glycolysis, we are gonna write on this piece of a paper down. I hope you have learned pretty much about how to make now a chart for the glycolysis cycle. Now, if that is messy, make a new one in the, maybe after coming from the college, that way you can memorize it right away, but do it in the same day. Okay, don't take it to the another day, otherwise you will, this entire thing will be getting erased, right? So that is pretty much it about the glycolysis. I wish you a very good luck, right? I wish you a very good luck, and bye-bye.